Our next lab is about enzyme activity, and as usual we're going to cover the important terms first. There should be quite a bit of overlap with material that was covered in lecture as well, but so let's take a look at our first term here is catalyst. A catalyst is something that speeds up a reaction without being used up in that reaction. And so in that sense, enzymes are biological catalysts. They will speed up the rate of a reaction without being used up as part of the reaction. Denaturation, by contrast, is when, for example, an enzyme loses its three-dimensional shape and it doesn't work anymore. So denaturation usually involves proteins, and it involves loss of activity because they lost their three-dimensional shape, which is critical to their function. Most of the reactions that we'll be having in this lab are digestion reactions, and they are basically hydrolysis reactions. So when you are eating food and you're breaking down these macromolecules into smaller building blocks, and that's referred to as hydrolysis. For example, when you're breaking down starch into simple sugars, that is hydrolysis. You're adding water into the reaction, and that breaks up these macromolecules into smaller subcomponents. In this case, it will be simple sugars. Dehydration synthesis is exactly the opposite. That's what happens when you're eating a lot of sugars and you don't use them up for energy. Then your liver will connect these simple sugars to make glycogen and then put it away for storage. So that means now we're removing water and building larger macromolecules by connecting these simple sugars together and forming starches such as glycogen that is then put away for storage. An enzyme is a biological catalyst, and it speeds up a chemical reaction. It's very specific to its substrate. So the general equation that we use for an enzyme reaction goes as follows. So the enzyme binds to the substrate, and this is a very specific kind of interaction. Every substrate has its own enzyme, and then it forms what's called an ES complex. I put it here with an asterisk because it's a rather unstable kind of configuration. It's relatively high in energy. And then at this place, the new bonds are being formed, old bonds are broken, and in the end, we're going to release a product, and the enzyme is recycled and then grabs on to the next substrate molecule. So in general, that's how an enzyme reaction works. So the terminology here, the enzyme substrate, and the place where the substrate binds to the enzyme, that's known as the active site of the enzyme. There are also modulation sites. These are different places on the enzyme. If, let's say, something like an allosteric inhibitor binds to a modulation site, then it could very well affect the active site of the enzyme and render it useless, basically. So a allosteric inhibitor would bind to a different site on the enzyme, and it changes the active site, and then the substrate doesn't fit anymore. So there are modulation sites. In the end, after an enzyme reaction is done, we're releasing a product, and that's shown here, abbreviated as P. So for the most part in this lab, you're going to have hydrolysis reactions. So these would be the typical digestion reactions, but there's also one redox reaction where we're not adding or removing water, like in a hydrolysis reaction or dehydration synthesis, but now we're moving electrons. And that's what happens in the Krebs cycle. We're following one specific type of reaction in the Krebs cycle, and that will be the succinic acid dehydrogenase, as it is, well, removing hydrogen and with that the electrons from the succinic acid, thereby oxidizing the succinic acid to fumaric acid, and we'll talk about it in a bit. Now, next up here on our list of terms is an inhibitor. Inhibitors can either be competitive or non-competitive. Competitive means that they're competing directly with the substrate for the same binding site on the enzyme. So a competitive inhibitor in general is very similar to the substrate. It's sort of a false substrate. It's a substrate in disguise. It fits into the same binding pocket that the natural substrate would fit into, sometimes even binding tighter than the natural substrate to the enzyme. And it's blocking that site for the substrate, and then the substrate doesn't fit anymore, and so you get no product. A non-competitive inhibitor is one that binds to the enzyme at an allosteric site, so somewhere else, not to the active site. So it doesn't directly compete with the substrate for the active site. However, the binding of a non-competitive inhibitor makes it so that the enzyme changes its shape, and with that shape changes the active site, and now the substrate doesn't fit. The natural substrate can no longer bind to the enzyme, and so therefore you also get no product. Both competitive and non-competitive inhibitors, they can be reversible or irreversible, meaning that they either permanently bind to the enzyme and they won't come off, or reversibly, that means depending on concentrations of the inhibitor, they reversibly bind, that means they will come off in the end. Our next pair of terms is coenzyme versus cofactors. Coenzyme, generally speaking, is a organic molecule, usually a vitamin, that helps the enzyme do its job. So it's usually required to be part of the enzyme reaction, and it does a job for the enzyme. For example, NADH and FADH, they both are coenzymes, so they're in the B vitamin group. Both of them, they help with energy metabolism. The organic molecules, they're dinucleotide, like the name indicates. So NADH stands for nicotinamide adenine dinucleotide. So the D is a dinucleotide. So two nucleotides together makes the dinucleotides. Is a not too large, but considerable still in size, an organic molecule composed of two nucleotides, like the ones that you would find in DNA, RNA. So adenine dinucleotide, flavin adenine dinucleotide, also dinucleotide based on the adenine base. But at any rate, so those would be both coenzymes. Now, a cofactor is generally an inorganic, typically a metal ion that is necessary for certain enzymes to occur, such as zinc or cobalt. If you're taking a look at your vitamin bottles or supplement bottles, they usually have a list of some metals that are necessary for certain enzyme reactions to occur. That would be considered a cofactor. So it's usually a metal, inorganic kind of factor that is required for the enzyme reaction to occur. Okay, next set of terms would be optimum, maximum, and minimum conditions. So optimum conditions are the best conditions. That is when the enzyme has its highest rate of product production because it has the best temperature, the best conditions for it to work. Maximum conditions are the maximum conditions to produce the maximum amount of product, and minimum conditions are the conditions that have to be met in order for this enzyme to work at all. You don't get much product in minimum conditions, but you get some. Okay, so let's take a look at the specific enzyme reactions that you are supposed to be doing in this lab, and of course it's going to be just all theoretical. It's a shame because this is actually a fun lab. It's one of the few very real wet labs that we have in physiology, and you're just going to get to sort of do it theoretically, looking at some images of the results. But for whatever it's worth, you should know these digestion reactions. They will also be later important when you do gastrointestinal physiology. So our first enzyme that's on the list here is amylase. And amylase is an enzyme that digests starch. So the general reaction goes as follows. So amylase is the enzyme that will digest starch down to simple sugars. And so I put the enzyme up here in red. Amylase will do that. Lipase is a fat digesting enzyme. And so your typical dietary fat, those are all triglycerides. So a triglyceride is being digested by lipase down to glycerol and fatty acids. So fatty acids are being released. Pepsin is a protease. It will digest proteins. And so here in general, a protein is being digested down to amino acids eventually. So it doesn't go in one step. There's quite a few processes in between. But pepsin is an enzyme that is found in your stomach, and it likes to have a very acidic pH for it to work well. So we're using pepsin to digest protein down to amino acids, actually smaller peptides first. And so you need quite a few proteases, peptidases. There are a number of them that are necessary in order to digest proteins literally down to single amino acids that can then be absorbed. So you need amino peptidases and carboxypeptidases and a whole host of proteases, such as trypsin, chymotrypsin, those kind of things. Anyway, for moving back to the fat digestion, the fats are kind of a problem for your body because fats are not water soluble, and all of these enzymes, they're proteins and they are water soluble. So how are you gonna use a water soluble enzyme to digest something that doesn't want to dissolve in water? Well, your body uses a trick. It's breaking down the fat into smaller droplets using emulsifier. The emulsifier that we have in the human body are bile salts, bile juices, bile salts, and they break down the fat into smaller droplets, which increases the surface area, and that makes it easier for your lipases to work and digest the fats. So emulsification, that's the process that we use to emulsify or keep these fat in smaller droplets. Renin is a substance that will convert the milk protein, which is casein, into an insoluble calcium perocaseinate. And if you ever heard the little kids rhyme, Little Miss Muffet sat on a tuffet eating her curds and whey. Well, if you use milk and you add renin to it, then the renin will make the milk into this semi-solid yogurt type of consistency. It will also produce a little bit of the whey, which is the liquid portion, but the curds, that is sort of this semi-solid yogurt type of consistency that you get after treating the milk with renin. Hydrolytic or digestive enzyme, that should probably be back up on the top there, but that's the type of enzyme. It will it will add water. Hydrolytic means we're lysing a larger molecule with addition of water. So hydrolytic means in this case, we're adding water, we're breaking down these larger macromolecules, say starches or a triglyceride or a protein, into simple building blocks such as simple sugars, glycerol, and fatty acids, and the amino acids or smaller peptides. So that's all hydrolytic or digestive. Now we do have one reaction here that removes actually hydrogens from a substance, from succinic acid, and so therefore this does not involve the addition or removal of water. So it's a different type of reaction. It's a redox reaction where we're removing hydrogens from one place and then putting it on another place. So in the Krebs cycle, you have a set of oxidation reactions. And so an oxidase is an enzyme that oxidizes something. A reductase is an enzyme that reduces something. So all of this involves the moving of hydrogens. So if we are oxidizing something, it doesn't necessarily mean that we're adding oxygen. It just means usually that we are removing hydrogen. So there are two definitions for oxidation. One would be to add oxygen, and the other one would be to remove hydrogen. In this case, we're removing hydrogen. Reductase is the opposite. We're adding hydrogen to something or removing the oxygen, conversely. So, but in our case here, our reaction just deals with hydrogen. Okay. So a dehydrogenase would be an enzyme that removes hydrogen. And so that's exactly what we have in this lab. It's a succinic acid dehydrogenase enzyme that removes hydrogens from succinic acid, thereby oxidizing the succinic acid. Polymer versus monomer. So all of these macromolecules, they're polymers of some sort. And here's as an example, let's use the carbohydrates here. Simple sugars are the building blocks. If we make a polymer of simple sugars, we get starch. And of course, if we are making a polymer of amino acids, we get a protein. So that's polymer versus monomer, where the amino acid would be the monomer and the protein would be the polymer of amino acids in this case. For fat, the situation is that we have triglycerides, which are broken down into glycerol and fatty acids. So the polymer would be the fat, or the triglyceride. The monomers would be the glycerol and the single fatty acids. For protein, that's the polymer, and the monomer would be the amino acids. And for starch, that's the polysaccharide, and then the glucose, that's the polymer, and the glucose would be the monomer. Absorption is what your body wants to do eventually with all of this, and pH is something that is the concentration of hydrogen ions.